Thank you for joining us today. We will begin in just a few minutes. For those of you just joining us, we will begin in a moment. Welcome. My name is Piper Chapman, and I am the Assistant Director at Harvard Alumni Travels. Thank you for joining us today for our travel talk. Before we get started, I'd like to briefly cover a few housekeeping items. I know many of us are quite familiar with Zoom webinar. However, for those of you joining us for the first time, I'd like to mention a few technical observations to best prepare for this lecture. Throughout the talk, your microphone will be muted and your video will be turned off so we can direct our attention to our speaker. However, the chat feature at the bottom of your screen will be on and accessible throughout the lecture. So please feel free to include your comments in the chat box and your questions in the Q&A box. We are offering live closed captioning during this lecture. For those of you interested in using this feature, please click the CC button at the bottom of your screen. Our webinar, which will be recorded, will last 1 hour, including time for Q&A. At the conclusion of our program, a very brief survey will appear. We'd be grateful if you would take the time to complete this survey so we can continue to better serve you. And finally, while we hope this lecture will run smoothly, we could experience glitches or delays in speech due to the constraints of technology. We appreciate your patience and understanding should this occur. If you are having Zoom technical difficulties, please note this lecture is being live streamed to YouTube, so you are welcome to join us that way. Please find the YouTube link in the chat. My favorite way to start our talk is by inviting all of you to enter your location in the chat so we can see where we're all tuning in from. So let's take a look. Great, thank you for sharing. It looks like we are joined by over 200 of our closest alumni and friends today, so thank you again for being with us. Now, to our Travel Talks lecture. It is my pleasure to introduce Hoekstra, the Alexander Agassiz Professor of Zoology at Harvard University and Curator of Mammals in the Museum of Comparative Zoology, amongst other university appointments. Hopi's research focuses on understanding the evolution of morphological and behavioral diversity in mammals, from identifying genes contributing to coloration to those that affect complex behaviors. Her research has taken her around the globe, including studies of grizzly bears in Yellowstone, rodents in Chile and Argentina, and mammals in the Kuriles Islands. We are so fortunate to have her with us today. So let's turn things over to Hopi to start her lecture titled Nature's Palette, the Evolution of Animal Coloration. Over to you, Hopi.
Great, thank you, Piper. Give me a second. Wonderful. So first I'd like to say thank you to all of you for joining in as we discuss what I like to refer to as nature's palette, how animal coloration has evolved over time. So today what I thought I would do is start by describing the process of natural selection. Because it's within that context, we're going to focus on two main questions. That is both why and how animal coloration has evolved. Now, because this is a Harvard alumni travel talk, I was inspired to focus primarily, although not exclusively, on African mammals, in part because they arguably are the most striking and iconic colors and patterns that we see in nature. So as Piper mentioned, I'm trained as an evolutionary biologist, so I'm hard-pressed not to start a talk without mentioning Darwin. So Darwin, what I find so remarkable about him is how much he got right even over 150 years ago. But Darwin sometimes is given credit for more than he's actually done. For example, many people believe that Darwin was the one who first came up with the idea of evolution. But in fact, the idea of evolution, the idea that populations change over time, preceded Darwin by over a century. Darwin's main contribution, which is arguably more important, is providing a mechanism by which evolution happened, that is, by natural selection. And it was in his most famous book on the origin of species, published in 1859, that he outlined this profound and elegant idea. So let me first describe the main tenets of evolution by natural selection. So the idea is that if you have a population, and within that population of individuals, there's competition. Not everybody can survive to reproduce. And if there are traits that are variable among those individuals that affect their ability to compete, and if those traits are heritable, that is, can be passed on from parent to offspring, then that population should evolve by natural selection. Now, what was so important about Darwin's book was that he provided not only this explanation, this idea, but he also provided a lot of evidence. So through careful experiments and observation, he documented competition among individuals. He documented that individuals varied in traits and those contributed to differences in survival. But there was one piece missing in Darwin's theory, and that was the idea of heritability. Now, while he appreciated that traits could be heritable, just based on how offspring tend to resemble their parents, he didn't have a mechanism by which this could occur, because of course in 1859 this was unknowable. We didn't know about genomes and DNA. So I thought I'd start today by sharing a brief anecdote that links Darwin to that second great discovery, that is three-dimensional structure of DNA, providing a mechanism for heritable traits. Okay. So what you're looking at here, I don't expect you to read, but just to appreciate, this is Darwin's last publication. It was published in the journal Nature, still a prominent scientific journal today, just 3 weeks before he died. His paper is entitled On the Dispersal of Freshwater Bivalves. And really what it is a natural history description describing a specimen in which there's a clam, or a freshwater cockle as he described it, clamped to a leg of a beetle. So why was this published in Nature even back in 1882? Well, it turns out that this explained this big issue at the time of how these cockles could move from lake to lake over terrestrial habitat. But what's more relevant to today's story is how Darwin got his hand on this particular specimen. Well, it was sent to him by a young shoemaker by training, but an amateur naturalist who was living in the British Midlands. He sent the specimen to Darwin. They had a flurry of correspondence. And it turns out this young shoemaker's name was Walter Drawbridge Crick. And that may ring a bell to some of you, because nearly a century later, that shoemaker's grandson, Francis Crick, with his colleague Jim Watson, former Harvard professor, and key but often unrecognized insights by Rosalind Franklin led to the discovery of the three-dimensional structure of DNA, how DNA can replicate and how traits can be passed on from generation to generation. What Jim Watson humbly referred to as the secret of life. And it's really in this DNA code that we find even more evidence for evolution by natural selection. Our 3 billion year existence, the shared evolutionary ancestry of all living things, and how changes in this DNA code can give rise to differences among individuals, populations, and species. So today, inspired by Darwin, we're going to explore the question of how this diversity arose. But thanks to Watson, Crick, and Franklin, we're going to use clues in the DNA code. So in particular, we're going to focus on these two questions. Why do traits vary, and how do traits vary in nature? So I'm going to frame today's discussion by considering the linkages between genes, traits, and the environment in which individuals find themselves. And as promised, the trait we're going to focus on today is color. So we're going to ask why color varies and how that's related to the environment in which a species finds itself, and how at the genetic level, changes in DNA give rise to the differences in color. So we're going to ask why color varies and how colors varies. Because in doing so, we can get a much more complete picture of how organisms adapt and why color has evolved. So why study color to begin with? Well, as you all know, color can vary tremendously among individuals, among species. It's one of the primary ways in which organisms interact with their environment, and it's involved in a number of biological processes. So we can all think about the iconic peacock's tail, how a male throws up its tail, flashes those brilliant colors to a female in hopes of wooing her to be his mate. The botanical equivalent of that is a flower petal color, where the colors of the petals attract pollinators. Those pollinators come and land on the flowers, attracting those pollinators to pick up pollen, bring them to another flower, thereby allowing plants to reproduce. But it's not all about attraction. Colors can be used also to warn other species. For example, in this poison arrow dart frog, the bright orange color suggests that this frog is toxic. It warns its predators away. But all good biological stories also have cheaters. So we know many examples of mimicry where individual species, they themselves don't bear the cost of producing a toxin or a venom, but mimic the patterns of those that do, and thereby receive the benefit of being less appealing to predators. So my favorite examples include the coral snake and its mimics, milk snakes and king snakes, or the iconic monarch butterfly, which is mimicked by the viceroy. And then a final example of many is crypsis. So here you're looking at a frog sitting on a log. If it's hard to make out the frog, well, that's the point. It's camouflaged in its local background. But the other reason color makes a good system to study is because we can take advantage of nearly a centuries of work done by geneticists who largely work on laboratory populations of mice. And in their giant colony of mice, let's say of white mice, every once in a while, there'll be a spontaneous mutation occur and a mouse is black. Then using genetic approaches, we can find and localize in the genome those genes that contribute to color differences. And these genes are largely conserved across mammals and mostly across vertebrates. So here I've just put up a list. Again, you don't have to look at the names of these genes, but just appreciate we know a lot of genes that affect color, how they interact, and how they work through development to produce color and pattern. So today we're going to start with the question of why color varies and how that's related to the environment. And so I thought, because of this is a travel talk, we could focus on one of the most mysterious questions, and that is why zebras have stripes. So this is a question that has long fascinated biologists, and there have since been, over the century, many hypotheses. So, for example, it's been posited that these stripes are actually important for camouflage. And when you look at a zebra, like this one in this picture that lives in a brushy habitat, the stripes actually help it blend in and break up the outline of the body of the zebra, therefore making it less obvious to predators. Another hypothesis is that the stripes cause confusion. Zebras often are found in herds. As zebras flee from a predator, the stripes confuse predators and they can't focus on any one individual, a process called motion dazzle. But maybe it's not all about predators. Another idea that surfaced is perhaps these stripe patterns are important for individual recognition. Because the stripes can vary among individuals, it allows individuals to use these visual cues to identify each other. Or it could be temperature, that the stripe pattern is important in convection cooling, allowing these zebras to thermoregulate in their habitats. And then finally, it's been thought that maybe they're important for reducing parasite load, in particular biting flies that may be affected by the stripe pattern. Now, the question of zebra stripes has not been sort of limited to a small group of scientists, but instead has captured the attention of the public. And in particular, there was an active debate between two players in the early 1900s. One of those players was Abbott Thayer, who was a well-known American painter, most famous for his cherubic paintings of young children. But in 1909, he published a book called Animal Coloration, in which he posited that animals vary by color, vary in color due to natural selection, but the mechanism was always camouflage. And he used his artistic skills, as shown here on this slide, to demonstrate that even the bright pink color of flamingos, if you observe that color at the right time of day, at the right perspective, may be camouflaging as they feed at sunset. He also weighed into this discussion about zebras, again suggesting that these stripes were camouflaged to zebras, especially if you looked at this from the perspective of an encroaching lion down on its belly, approaching a potential prey. But many in the community believed that Thayer took this too far, that it wasn't all about camouflage. And one of the people who was particularly vocal in this debate was Teddy Roosevelt. He had stepped down as president and just returned from a hunting expedition in Africa. And having seen zebra in the flesh, argued that there was no way these stripes could be important for camouflage. If anything, it made the zebra more conspicuous. He, along with, at the time, two Harvard MCZ curators, argued that color could be used for a number of different reasons. So for example, this lilac-breasted roller, certainly those colors weren't camouflaging, but could be important for things like sexual selection, that is, attracting mates. So these stories by Thayer and Roosevelt and the ensuing back and forth, strongly worded debate that followed were just ideas. They really didn't have experimental evidence. So one could argue that they were reminiscent of the Just So stories put forth and written by Rudyard Kipling in 1902, where he had these fanciful tales about how the camel got its hump, the leopard its spots. You could add another chapter about how the zebra got his stripes. If we really want to understand how zebras got their stripes, why they varied in color, next thing we needed to do was really do an experiment. So I would argue that over the last couple decades, we're starting to hone in at least on one reason why zebras may have evolved stripes. And that's this idea that in fact, stripes might be important in deterring parasitic flies. Now, there were two studies early on that I'm going to mention that set the foundation for some recent experimental evidence. The first note was that there's a strong correlation between the distribution of horseflies, these biting flies, and striped horses, including zebras. That is, wherever there were horseflies, the equids, members of the horse family, were striped. And in areas of the globe where there were no biting flies, the horses were solid in color. So that was a tantalizing correlation. And this was followed by an experiment in which researchers put out sticky surface to capture flies that landed on it, but put out different versions of stripes where the stripes were wide or narrow, and looked at how many flies landed on those surfaces. And what they found was that flies landed less frequently on striped surfaces, and in particular, those stripes that mimicked, at least in size, that seen in zebras. So I think these types of studies laid the foundation for the key experiment, and that is doing an experiment in zebras themselves. And in fact, there were two studies just last year that approached this. And here's what they did. They used cows as their model, and they painted cows with zebra stripes, as shown in this middle image. As controls, they used a cow that was on the left, painted with just black stripe, and on the right, a cow that had no stripes. And they asked how many flies landed on these differently colored cows. And what they found was actually quite striking. That is, when a cow was colored like a zebra, fewer biting flies landed on them compared to either of the two controls. And it turned out that this was corroborated by another study published in 2019, which took a similar approach, but this time put coats on the backs of horses that were colored either like zebras or solid in color. So together, these studies really suggest that at least one of the reasons why zebras may have evolved stripes is because they deter parasitic or biting flies. Now, it's important to note that these experiments don't rule out the possibility that stripes are also important for camouflage or motion dazzle, but do suggest a role for natural selection in driving these differences. Okay, so now that we have some idea about why zebra stripes may have evolved, I'm going to turn to the question of how zebra stripes may have evolved. So I'm going to talk about two particular cases, one in which we see differences either in individuals or in regions of the body of a single individual, because in some regions there's pigment and in other regions they lack pigment. And the second case in which a region or an individual is completely pigmented, but they use different types of pigments. So to discuss these two things, I want to provide some background information about how pigmentation works, in particular in mammals. So pigment is produced by a specialized cell called a melanocyte. We'll call them pigment cells here. And pigment cells are found in a number of tissues in the body. Most notably, you can find them in the skin, in the eyes, and hair follicle, as well as other regions. But what's important is that the presence or absence of these pigment cells, the concentration of pigment cells or their activity can vary among tissue types. And that's why we can have people that have dark skin but light hair, or dark hair and light eyes. So you can dissociate those traits. Now for today's talk, because we're talking about mammals, I'm going to focus primarily on skin and hair follicles. So melanocytes are pigment cells that are in the skin that produce pigment that give us different skin tones, and those in the hair follicles that produce pigments that are then deposited on growing hairs. Okay, so in mammals, we produce just two types of pigment. One is called eumelanin and the other pheomelanin. Eumelanin, or dark pigment, is responsible for the colors brown to black, whereas pheomelanin gives rise to hair colors that are blonde to red. And the difference between a blonde hair and a red hair is simply the concentration of pheomelanin. And similarly, eumelanin is the concentration of eumelanin from brown to black hair. So here's where I'm going to invite you to pluck out one of your own hairs, and you can have a look at that hair. And you can determine whether you have eumelanin or pheomelanin. Now, my guess is some of you will do that, and you might come to the conclusion that you have neither of the two. And that's because potentially, as we age, the population of stem cells that give rise to these pigment cells starts to be depleted. So we're producing fewer pigment cells, hence less pigment, and that results over time with age, hair that looks white or gray. Okay, the other thing you might notice, besides the pigment type in your hairs, is that your hair is usually uniformly colored. That is, it looks the same at the tip and at the base. But this pattern is actually quite boring in terms of mammalian diversity, because most mammals can have hairs that are different in terms of their pigment types. So if we took a mouse, for example, or a deer or a grizzly bear, the most common hair pattern is one that looks like this, where we have eumelanin or black pigment at the tip and the base, and a pheomelanic or light band in between. And we can get different shades of bears and mice and deer simply by changing the size of that band. But some animals have even more complex banding patterns, where you can go from dark pigment to no pigment to light pigment to no pigment, etc. So now here's where I'll invite you, if you have a dog or a cat that has different colored hairs, maybe not pluck it, but you could look at those hairs and you can see extraordinary patterns of pigment or lack of pigment along the hair. Okay, so how does a cell know what type of pigment to produce? Well, pigment cells have a switch, and that switch is really, molecularly speaking, a gene called the melanocortin 1 receptor. When this receptor is activated, it signals through a signaling molecule to turn on the production of dark pigment. If that activator goes away or there are mutations in the melanocortin-1 receptor, then the signaling molecule goes down in concentration and you produce pheomelanin. So you turn on the switch and you get dark pigment, and you turn it off and you get light pigment. Okay, so the two things I want you to remember about pigmentation is, one, that pigment cells need to be present to make pigment. And two, if you have those cells present, a cell can produce either dark or light pigment. Okay, so first let's ask about the presence or absence of these pigment cells and how that mechanism might contribute to variation that we see in nature. So it's long been known that patterns like the ones shown here, the tuxedos of cats or the blaze on a horse, are caused by the lack of pigment cells. So pigment cells arise in the neural crest, that is along your back, and during development they migrate through your skin around and connect at your midline or your belly. So when you see a pattern like this, it's often caused simply by the migration of those melanocytes not being complete. They don't make it all the way around. So because of that, you can see a blaze that often happens in the midline on the head or the midline of a cat. So this is a pattern that's generated by the different distribution of pigment cells. And although this is not known, it's kind of tantalizing to think that this type of mechanism may give rise to some of the patterns that we see, for example, in African ungulate. So you see this bontebok, which has a blaze that's highly reminiscent of a horse blaze. Okay, so let's now return to talking about zebras. How does the presence or absence of pigment cells contribute to zebra coloration? Well, it turns out that zebras have pigment cells, and they're all across the body. So in other words, if you shave the fur of a zebra, the skin is completely black. And we already got a clue of that because every once in a while you see in the news there's a mutant zebra that appears. This one is taken from Kenya. And this is the zebra that has, instead of stripes, it has spots. So those stripes are broken up. But you can see in those other areas that there are, the fur is dark. But here's what becomes interesting with zebras is the reason they have white hairs on those black skins and the striping patterns is because in the formation of stripes, the stripes that are black, there are pigment cells in those hair follicles, and so those hairs become pigmented. But in the white stripes, there are no pigment cells in the hair follicles, and so you get unpigmented hairs which appear white. So this is the mechanism by which we get those differences in color. But of course, the next question is, how do they get those colors in the stripe pattern? Well, let me tell you right now is that we don't know the answer, in part because it's very hard to study development in zebras. But I'm going to tell you about a hypothesis that explains some of the thinking about how we could get these stripe patterns and variation in the patterns. And this comes about through the observation that if we look at the 3 species of zebra that occur in Africa, they all have stripes, but their stripes differ in a consistent way. That is, the plain zebra tend to have relatively broad stripes. By contrast, Grevy zebra tends to have much narrower stripes. So if we think about how you might affect the banding pattern or the striping pattern, here's the leading hypothesis. So imagine, so when a zebra is born, it's born with its stripes already intact. So what that means is that as a developing zebra gestating in its mother, this is when stripes are made. So now imagine this little zebra embryo being a little balloon. And if you're going to draw stripes on that balloon, let's say 1 centimeter in width, and then as you blow up that balloon, as the zebra gets bigger and bigger, you're going to end up with fairly broad stripes. But if you just delayed the time in which the stripes were set down until the balloon was medium size, and you again make 1 centimeter white stripes across that whole balloon, it's going, as the balloon gets bigger or the embryo gets bigger, the stripes are going to be more narrow. So the thought is that the reason that we get these different patterns across zebra species is simply a matter of timing when the developmental program starts to put down those stripes. This, of course, leaves the question is how do we lay down stripes? But that's at present an unanswered question. Okay, so this is a mechanism by regulating where pigment cells are. We can generate patterns, but we can, as I mentioned, we can also turn to the question of how can we change the type of pigment that's made? And I already alluded to this switch, which is the melanocortin-1 receptor. This particular switch has been the subject of a lot of genetic studies. So here's the structure of it in cartoon form. The main thing to note is each of these circles is an amino acid that together make up the protein. It's found on the membrane of these pigment cells, and its job is to take information outside the cell and transfer it inside the cell. And all of the circles that I've colored in are known mutations that affect pigmentation. But I quickly just want to tell you about one of these mutations. It's in the 65th amino acid. It changes an arginine to cysteine. The details are not important. But we discovered this mutation in our own research as we had been studying the mice that occur in Florida's Gulf and Atlantic coasts. So on the right, what you see is a typical mouse living in mainland Florida, let's say, in these dense fields with dark loamy soils. They have a dark colored back, a striped tail, and a dark face. But what we found is we moved to the beaches off Florida's beautiful Gulf and Atlantic coasts on these barrier islands. If we catch the same species of mouse, as you can see in this image, it looks quite different. It lacks pigment on its flank, its tail, and its face. And one of the contributing changes is this mutation in the melanocortin-1 receptor. So here we've been able to connect a mutation to differences in color and have done experiments to show that color matters in these natural populations. But what was even more remarkable about this study is that the same mutation was found in another species of mammals. So if you just sit back and think about all the mammals that you can remember, and you start to think about what mammal is most different from a mouse. So here's where we can do audience participation. If we were live, I would probably hear things like blue whale, and once in a while I hear elephant. And what I'm going for is something close to elephant, and that is a mammoth. So about 15 years ago, maybe 20 years ago now, researchers were really keen on being able to sequence DNA from an extinct organism. So the first thing they had to do was pick an organism, and they chose mammoth. And mammoths are a good organism to sequence ancient or extinct DNA for two reasons. One, because they're only about 15,000 years ago they went extinct. It's not that long ago. But second, their bodies are often, and bones are often discovered in the permafrost in northern Siberia. So this is like keeping your DNA in a freezer, right? So it's kind of the best conditions you can imagine for keeping that DNA intact. So when researchers decided that their goal was to sequence a whole gene from an extinct organism, they chose mammoths, and the gene they chose was this melanocortin-1 receptor, in part because it's a simple gene with a short sequence. And when they did so in 3 individuals, what they found was, at least in one of those individuals, there was a mutation. And it turned out it was the same arginine to cysteine mutation at position 65. And based on our work in mice, what this suggested is that mammoths, like mice, may have been polymorphic in color. But of course, in this study, we could return to the question of why did mammoths have different colors? And all I can say is that when this study was published, the newspapers read, blonde mammoths have more fun, suggesting that may be important in mate choice. But of course, that's pure speculation. And if you thought doing experiments in zebras were hard, they're even harder in mammoths. Now, while this is a case in which you have the same gene and the same mutation, as I mentioned earlier, there are other mutations in this melanocortin switch gene that give rise to differences in color in a whole diversity of vertebrates. So for example, in this particular breed of cattle, a mutation in the melanocortin-1 receptor will take what would be a black cow and give it this beautiful auburn color. For those of you who are dog lovers or maybe even have a Labrador at home, if you have a golden lab or a black lab, the difference between the two is a simple single mutation in the melanocortin-1 receptor. This also occurs in lizards. So I think you can look at these pictures of these two lizards that represent the same species, but one occurs in the deserts of New Mexico and the other on the brilliant white gypsum sands of White Sands, New Mexico. And I bet you can pick which is which. Well, what about us, humans? Well, it turns out that there are mutations or variants of MC1R that occur in human populations. One of the most notable is a mutation that leads to light skin color and auburn hair. This is particularly at high frequency in human populations and in Ireland in particular. Now, as many of you may know, in recent years, the Neanderthal genome has been sequenced. And in doing so, of course, we can look at the melanocortin-1 receptor. And what researchers noticed is that there's a mutation in the MC1R gene. Now, while we usually think of Neanderthals and they're usually depicted as having dark hair and dark skin, what this study suggested is about 5 to 6% of Neanderthal populations, individuals, likely had red hair and light skin. Now, it's interesting to note, it's not the same mutation found in humans, and that's important, one, because it suggests it's not simply human contamination of Neanderthal DNA, but it has the same effect on the MC1R structure and function, and hence likely to lead to this light coloration. But I don't want to leave you with the impression that all variation in skin and hair color is due to melanocortin. It's one of just a few genes that have been identified in human populations. There's a growing list. So you may be wondering, how come we know so much about cattle coloration and even zebra coloration, but we know so much less about the genetics of human skin color? Well, it turns out we've learned a lot, as I alluded to earlier, about skin color, hair color, excuse me, from mouse populations. Well, it turns out mice are a great model for hair color, but not a great model for skin color, because mice don't have any pigment cells in their skin. So if you take a black mouse, a brown mouse, a white mouse, shave their hair, they're all pink underneath. And so many of the genes that we now know contribute to human skin color variation were first discovered in other organisms. So for example, the second gene listed here, the KIT ligand, was discovered in fish and then later studied in humans. So what I've hoped to convince you of today is that by understanding both why color varies and to give you a sense of, while it's easy to come up with lots of hypotheses, it's harder to do experiments. And sometimes when you do the experiment, you come up with a surprising result. But combining that with understanding how color varies, we have a much more complete picture of the evolution of color, both within and between species. But of course, there are many unanswered questions, so I just want to mention two things that I'm particularly excited about. The why questions, one on the why question and one on the how question. So there's so many unanswered questions about why animals vary in color, but I thought I'd just mention one that just was reported in the news last week, and that was the observation that platypus glow. As if platypuses weren't unique and weird enough with their duck bill, they produce venom, they have, males have spurs on their heels, they lay eggs. Well, it turns out also under UV, they glow. And this came about by a simple chance, where a forestry professor was out in the woods with a UV light and happened upon a flying squirrel, shined it on this squirrel, and the flying squirrel glowed this bubblegum pink color. And that led them to then go investigate collections of museum specimens and to find that other species of mammal glow as well. And when this was reported in the New York Times, the headlines read, "Platypus glows, but we have no idea why." So I think this is going to be a fun puzzle to try to answer. But we also want to know other questions on the how side of things. And one I think of the outstanding question is, how do animals get not just differences in color, but these complex patterning differences? And I think we have a lot of inspiration to think about things like stripes coming from zebrafish, and they're zebra mice. So these are mice that live in the grasslands of Northern Africa. And we've contributed a bit to this research by studying this African striped mouse found in South Africa, in which we learn the molecular developmental basis of their stripes that they form on their back, these longitudinal stripes, which also give rise to the same types of stripes, but independently in chipmunks that we have here in Massachusetts locally. But I think there's a lot to learn about how these more complex patterns are formed in fish, in birds, and in mammals. So with that, let me bring it back to the start of the talk where I mentioned Darwin, just to end by saying, I hope if Darwin was alive today, he would be tickled by all that we've learned since his time in particular, not just about why animals vary in traits and in particular color, but how, by studying their genetics, that missing piece to Darwin's great theory of evolution by natural selection. So with that, I'm going to end here. Thank you for your attention. I'm happy to answer questions. And I didn't want to leave you hanging in the sense that I showed this image at the very beginning of the talk. So I wanted to provide you the answers to what each of these color patterns was. So here are those answers. And just to note, these are the species that we saw on the last Harvard alumni trip I took to Africa last year. Thank you.
Thank you so much, Hopi. This was wonderful. I'd love to transition to the Q&A portion of our program. I should first mention that we received over 40 pre-submitted questions, so we have grouped some of them based on similar topics and themes. We'll do our best to get to as many as possible. As we begin with a question on animal perception, we're going to leave this final slide on your screen so you can spend some time analyzing or enjoying or matching some of these beautiful patterns. So let's go to our first question, and that is, how do animals' perception of color differ from that of humans?
Okay, this is a great question. So I talked all about color and color patterns, but of course the sort of second flip side of that, or that's equally important, is how those color and patterns are perceived. So humans in general are not that, depending on what you compare us to, are not that good at perceiving color, at least compared to some animals, but we're better than others. So color perception is really driven by cones and the opsin genes in those cones. Humans are considered trichromatic. So we have 3 different receptors that cover 400 to about 700 nanometers in terms of wavelength. And so that's how we perceive the world. But of course, there are some animals that also have a 4th cone, and that generally allows them also to see colors in the UV. So, you know, there's questions about that UV coloration when we talk about platypus, you know, and the question of, can platypus see that coloration? That being said, there are many mammals that don't perceive color as well as we do. So for example, rodents and dogs tend to have 2 opsins, 2 cones. And so they see the world much more in less coloration than we do. And most aquatic animals, or mammals, I should say, only have 1 cone, so they see the world much more in a grayscale. So in some cases, we definitely see the world differently. And that's important when we're thinking about the study of color, because the way we perceive color is different. Whereas some vertebrates see color better than we do, others less so.
Great, thank you. Our next question, can you explain the annual extreme changes in coloration in some species?
Okay, so I think this is a really exciting topic and quite timely in the sense that there's been renewed interest in these seasonally changing animals. So things like hares, weasels, hamsters is another one, foxes, some birds like members of the grouse family. They change their color seasonally, so they tend to be white in the winter and more brownish mottled color in the summer. This has been well studied, certainly in some species, and it's pretty clear that at least one of the factors driving that switch in coloration has to do with camouflage. And that could be camouflage against predators or camouflaging yourself if you are a predator so that prey are less likely to see you. And because there's been a new interest in this in part because of climate change is affecting the timing of these switches and generating at some times of year a mismatch between animals and their environments. But what I think is really interesting is two things. One, it's pretty clear that the change is driven primarily by hormones, a hormone signal that starts this change in color. But even more recently, folks have been able to nail down some of the genetic changes that may be involved. And one of the key genes that seems to play a role in seasonal color change, at least in mammals, is a gene called the agouti signaling protein. And interestingly, it is the gene that interacts physically with the melanocortin-1 receptor that switches the pigment back and forth. And so there's a clear mechanism there of how they change color, at least at the level of pigment.
Very interesting, thank you. So our next question here, could you touch upon the biochemical pathway and evolution behind birds of colors? For example, parrots and birds of paradise.
Yeah, so sometimes I because I study color in mammals, sometimes I get a little teased in the sense that mammals really come in, white to light brown to brown and sometimes black. And certainly not as much color variation as we see in other groups, namely birds, which have these brilliant colors and arguably more interesting patterns, I loathe to admit. So birds, like mammals, have these melanins. They produce eumelanin and pheomelanin, and the mechanism is largely conserved. But what birds have is they have sort of additional pigments. They have pigments like carotenoids that they ingest in their environment. This is probably best illustrated by house finches, which are common. You may see them in your backyard, for example. The red chest that you see on these house finches is largely just the intake of carotenoids that are then incorporated into developing feathers. But you also notice on birds, there are other types of pigments that are thought to be other types of colors that are not caused by pigments themselves, but instead are considered structural colors. So these can be the iridescent colors that you see on the throat of a hummingbird. So you look at a hummingbird and you can see, depending on the angle at which you look at, that sort of brilliant flash of color, important, we think, in mate choice. And those are caused, that color is caused by these microstructures on the little end bits of feathers. And so what can happen is the light comes in and gets, for example, reflected in different ways, giving that iridescent color. Then there's non-iridescent structural colors. So for example, the blue colors we see in blue jays, that's largely caused by little air pockets, again, at the end of feathers that scatter light and reflect in blue. And there's also UV scattering as well. So there's some melanin-based colors, other pigments, and then these structural colors, which there are some other mechanisms, but those are the main ways that birds vary. And there's some beautiful research going on bird coloration.
Great, yeah, thank you so much. We're gonna travel back in time for our next pair of questions here. So the first one, could you expand on dinosaur coloration?
Yeah, so I think dinosaur coloration is really fascinating. And in particular, in recent years, there's been some, I would say, big breakthroughs. So I was sort of surprised to learn that the images that we see in a lot of books and children's books depicting dinosaurs, those colors, the colors of dinosaurs is more or less made up. We had no idea what color dinosaurs are. So you see sometimes they're green and sometimes they're brown and so forth. But in recent years, there's been some clues that actually are based in reality. And that is because there have been specimens that have been identified where you can look microscopically at the structure of the skin. So remember when I showed you that picture of a pigment cell and I showed you eumelanin and pheomelanin? Not only were those circles differently colored, but they were differently shaped. And that's because these melanosomes that contain pigment are more round when they're light pigment and more oval when they're dark pigment. And so you can see this actually, if you look closely enough, in the skin of these fossils. And that has given rise to, not only do we have a better sense of the color, but also patterning. So there's some reconstructions of dinosaurs that are striped, that have different patches of color, that have, bright red crests, that are all being discovered just in the last, I would say, 10 years or so.
Interesting. I must confess I have a vision of dinosaurs being these green figures, so thank you for clarifying that. So the next question here, how have ancient cave paintings and paleontology informed our understanding of animal coloration? For example, spotted horses and dino feathers.
Great. I think this is a nice question that sort of follows up on this. Cave paintings, I learned, generally the pigments used are black, yellow, and red, and some white. So we're sort of limited in what colors are seen. But of the about 4,000 animals that are depicted in cave paintings, about a third of them are horses. And most of those horses are drawn in either black or brown. But there's a cave in southern France that dates back, the paintings date back to about 25,000 years ago, in which there are drawings of horses that are white with black spots on them. It had been presumed that these kind of interesting color patterns in domesticated in horses came about through artificial selection during the process of domestication, which occurred about 5,000 years ago. But this cave painting gave us this really clear indication that there were spotted horses long before they were domesticated. And I love that because it gives us a clue about something in the past that we wouldn't have known about. And our intuition that it was a process of domestication was in fact wrong.
Great, thank you. Okay, well, I'm particularly fond of this next question as it relates to these beautiful creatures in my background that we've heard so much about today. So why are some zebra stripes brown and others are black? And I do believe that one of the zebras behind me actually has brown stripes, which hopefully you can correct me if I'm wrong there, but I do recall seeing many brown striped zebras.
Yeah. And if I get this right, it's the broad striped common or plain zebras that also tend to have what are referred to as these shadow stripes. So in the white stripe neighboring the black, it's almost like a lighter colored stripe that often is more brown than the actual stripe. But zebras do tend to vary in the intensity of that eumelanin we see in the black or brown stripes. But unfortunately, I have to admit that we don't know the reason why. And again, we're just discovering why they have stripes in general. So here's a pretty powerful experiment, something that's striped and not striped. But knowing why these more subtle differences in black to brown, I think is going to be a really hard question. And it could be that it's not driven by natural selection. It's just that there's varying amounts of eumelanin, whether that's based on genetics or age. I should mention that it's known that young zebras tend to be more brown and they get darker through time. So some of the difference could be developmental over time. But I think it's a fascinating question. It'll be fun to learn the answer, but we don't know it at the moment, or at least I don't know it.
Great, thank you. And you have also touched upon this too, but what do stripes do for some animals?
Yeah, so today, of course, we talked about these sort of stripes that go from the back to the belly that we see in zebras, but of course there are many organisms that have stripes that are longitudinal, like go along the body. And I think that's especially common. And as I mentioned very briefly, we've been involved in some research trying to understand the stripe pattern in mammals. And in mammals, we know that those dorsal stripes that we see on chipmunks, for example, are very common in mammals that are diurnal, so active during the day, and are tend to be prey species. And this has evolved many times independently, and that suggests there's some function. So researchers have been trying to understand why. And so one of the clever experiments that researchers did is they had little objects move across a computer screen, and a human subject was meant to take their finger and try to tap that moving object. And it turns out that if you put longitudinal stripes on that object, we missed those moving objects more than if they were solidly colored. So that suggests that, in fact, that may contribute to an increased ability to evade predators if you have that stripe pattern. And that's also consistent with research that's been done in snakes. So snakes, right, well, some snake species have these longitudinal stripes and others are more blotchy, let's say like a rattlesnake. And those patterns are really closely correlated with escape behavior. So snakes that sort of hide and sit tend to be blotchy and more camouflaged, where those that when they see, a potential predator sort of slink away, those tend to have striped patterns. So I would say that's the leading hypothesis, and folks are designing experiments to test that.
Very interesting, thank you. We're gonna look more closely at color in our next question here. Is there any evidence that animal species color has evolved over time?
Yeah, so I think this question, to answer this question, we have to think about time, what the time scale is, because clearly patterns and color differences have evolved over the course of millions of years. We see that color evolves over sort of ontogenetic time within the lifetime, whether it's our gray hair or the zebras getting darker, et cetera. And we can think of lots of great examples of young animals that have completely different colors and patterns than their adult form. So one of my favorite example is the tapir that's got these beautiful striped patterns, and then as an adult has sort of a dark, light, dark pattern across its body, or fawns and of deer species often have these spots, and that has to do with their the way that they evade predators when they're young versus when they're older. But if we think about evolutionary time more over the course of thousands of years, I think there's some really striking examples. So one example is these beach mice that I mentioned on one of the slides that I presented today, where we have this dark mouse that lives in mainland Florida, but as they started to colonize these beach habitats, they have evolved this lighter camouflaging coloration for this novel environment. What's cool is we know something about the age of these sand dune islands on which these beach mice occur, and they're only about 3,000 years old. So the assumption there is that we've seen color change, although we haven't witnessed it over our lifetime. We certainly see that over just a few thousand years. And I think there's some really interesting studies that have used more evolutionary approaches to try to reconstruct, for example, what an ancestral species that no longer exists, what colors those are as well. So I think there's lots of examples, but those examples differ depending on what time scale you're talking about.
Thank you. So the next question that came in, I think this is really interesting. How much of color is used for competition versus reproduction?
Great. So today I focused a lot on sort of natural selection, but another component that's closely allied to that is sexual selection. So natural selection we tend to think of in terms of survival, and sexual selection we tend to think of in terms of reproduction. And both of those are important components to fitness. If you're thinking about passing on your genes, you have to survive long enough to reproduce. So the relative sort of weights of those two really is going to depend on the ecology of the organisms. So for example, in mammals, camouflage ends up being really important. And that's primarily, I would argue, because most mammals, I'm going to generalize, are nocturnal, and they tend to use olfaction as a sense of smell as a primary way of communication and recognizing each other and so forth. So in that case, maybe one would argue survival is more important. But birds, I think, are a completely different story, in part because they're diurnal, they're very visual, and so colors and patterns arguably may be more important for reproduction compared to at least mammals. But I think the important thing to remember is that for most organisms, it's really a balance between the two. So if you're colorful and you're advertising yourself to potential mates, that may come at a cost because you're also more visible to predators. Right. So the colors that we see in natural environments are really kind of a balance point between those two. How much benefit do you get from reproduction, and potentially how much cost to survival? So I like to think of that as more of a trade-off between the two.
Mm-hmm. Sure. Well, I think we have time for one final question here. And let's go to what are the biggest unexplained issues or questions related to evolution of color?
Yeah, so you're going to get my biased perspective on this, and I sort of gave you a hint. as to the kinds of questions that I think are largely unanswered. I think we're never going to run out of questions about why color varies. But as you sort of, I hope, got the sense of, answering that question can actually be very hard. Like, how do you design experiments to test this? And often we want to test this in these, exotic, brightly colored birds in New Guinea or zebras in Africa, and that becomes, has its own challenges. But I think we're just, like the example from the platypus, I think we're discovering these really cool new patterns that, in this case, weren't even visible to us. I think there's a lot to be done at the molecular level too. And in particular, while we are gaining a better understanding of differences in pigments, I think we know much less about patterns and complex patterns. And in particular, I would say in birds that, again, arguably may have more exciting patterns overall than mammals. So there are some really fantastic labs, including a lab headed by a former postdoc of mine, who's really looking at birds, and they're doing these great experiments in, for example, emus. Emus have these amazing stripy color patterns in young emus, and it's doing some beautiful work trying to understand how stripes are formed in those groups. And there's been some wonderful studies about structural coloration too, and I think there's a lot to learn, whether it's birds or butterflies, these beautiful iridescent bright blues and greens that we just can't study in mammals.
Thank you so much. Well, there have been so many questions, and unfortunately, we just don't have time to get to all of them. However, Hopi, on behalf of the Harvard travel team and our alumni community, I'd like to thank you so much for sharing your knowledge with us today. Hopi is scheduled to lead a travel trip next year, Family Tanzania Safari, from December 26, 2021 to January 6, 2022. I'm sure she'd be delighted if you would join her. Please feel free to contact our office if you have any questions. Finally, you will shortly receive a follow-up email, including a virtual guide containing the recording of this lecture, a reading list, and some other really neat resources. Thank you again for joining us today, and please stay well.